Welcome everyone to another SEGO seminar. I'm Robert and I'll be hosting. And so during the presentation, I would like to ask that everyone keep their camera off and stay muted. And if you have any questions, just type them up and message them to me as a private message during the talk. And at the end, we'll do a Q&A. And so I will pass the questions along for you at that time. So that way you don't have to remember for the entire talk. You can just message it to me and we'll take care of it. So I'd just like to pass it along to Dr. Holstein, who will introduce our speaker, Dr. Philip Gravenese, and take it away. Thank you.
All right, thanks, Robert. I'm very excited to introduce everybody to Dr. Gravenese, who does some really cool research on some very delicious crustaceans. Phil is an assistant professor of marine biology at Florida Southern College. He received his BS in marine biology in 2003 and his MS in biology in 2007 from Florida Institute of Technology. He then spent 6 years teaching as a professional educator in Florida's public schools before he returned to Florida Tech to earn his PhD in biology in 2016. During his PhD, not only was he doing his doctoral research, but he was also dedicated to educational outreach. He co-founded the educational nonprofit Youth Making Ripples, which challenges K through 12 students to create short ocean-themed documentaries that raise awareness about critical marine issues. This is a very cool program. I encourage you to look into Youth Making Ripples if you're interested. In 2017, he accepted a postdoctoral fellowship at Mote Marine Laboratory and Aquarium in Sarasota, and he was there until 2020 when he joined the faculty at Florida Southern College. His research is focused on determining how different environmental stressors like ocean acidification, elevated temperature, hypoxia, and red tide may impact the development, survival, and behavior of commercially important crustaceans, including the Florida stone crab and the Caribbean spiny lobster. And with that, I will hand it over to Dr. Gravenese.
Well, thank you everyone for coming today and for the invitation to be a seminar speaker for your lecture series. Really excited to be here today and share with you some of the work that I've been doing over the last couple of years throughout my postdoc and what I'm starting to do or continuing at Florida Southern College. So I grew up outside of Philadelphia, and I spent my summers with my grandfather down in Cape May, outside of Cape May, the Jersey Shore, and we were always fishing and crabbing. And this is kind of where my love for the ocean kind of was built around these experiences. And it was interesting. So even through my adolescence and into my teenage years with participating in these activities with my grandfather, we saw the blue crab, which was what we were catching, go through a drastic decline during the 90s. And so it was kind of serendipitous when I got to Florida Tech that one of my mentors was a blue crab biologist. And so I naturally gravitated towards his lab, and that's where as Dan mentioned, that's where I did most of my training. And so my research focuses on the different environmental stressors as a singular stressor, but also multiple stressors simultaneously. And we do different experiments to identify what the tolerance of these organisms might be throughout the different stages of their life cycle, and also how it might impact their behavior. So these are three of the main species I work with in Florida: the Florida stone crab, the Caribbean spiny lobster, and I recently started working with the Caribbean king crab as well. But today I'm just going to focus on these two stressors, ocean acidification and elevated temperature. And I'm also going to primarily focus on the stone crab and some of the behavioral work that we've done with the stone crab. And then at the end, I will talk to you a little bit about some really exciting work that we just completed with some of the larvae of the Caribbean spiny lobster. So this plot shows us the species contribution to the United States fisheries, and a lot of the fishery that you see here is about 50% is associated with those typical finfish that you would see if you went to a restaurant. But a lot of times folks tend to overlook or forget that 30% of our national fisheries is essentially dedicated to crustaceans, lobsters, shrimp and crabs across the nation. And so this plot was the latest plot that NOAA had that was present for the top 10 states for NOAA fisheries, and California is the first, but Florida is up there. It's number two. And over the last couple of years, from 2004 to 2015, in our crustacean fisheries, we've seen a 68% increase in the harvest in our crustacean fisheries. So they're popular, and we in Florida celebrate our iconic species. We have seafood festivals that are dedicated to the start of each of the fishing seasons for stone crabs and spiny lobster. They play a huge role in local economies and bringing tourism and people in to participate in these seafood festivals and celebrate the season. In the Keys, there's actually a very slow stone crab eating competition that happens right around the time of the season opening. And it's always a fun event to go to. So it's become part of, at least in South Florida, the culture to incorporate some of our iconic marine fisheries into our local day-to-day lives. So the stone crab, Nippy mercenaria is its name. It's the Atlantic stone crab. This is what they look like, but it's a unique fishery because the fishery is associated with removal of the claw and the animals returned back to the ocean. And so that brings up a couple of issues with kind of monitoring the fishery, because it's not like a blue crab where an individual is being harvested completely. And it's thought that the claws will regrow and they'll regenerate those claws and in a couple of years re-enter the fishery as a secondary harvestable organism. It's, like, as I mentioned, it's economically important in Florida. Annually, it contributes about $30 million to our local economy, and we will actually ship out a lot of, and that's just the recreational fishery. We'll ship a lot of these claws out to other states and even other countries. This is the commercial fishery. The recreational fishery is not included in this value, and it's a little bit harder to track the recreational fishery. So over the years, we've seen in the press articles like this: where have all the stone crabs gone? Shortages in Florida drive up the price. In December 2017, we had another article that came out talking about the shortage that was happening. And I just want to kind of highlight the fact that you can go back, you could do a Google search, and you can go back in time, and these events of shortages are more common in the last 20 or 30 years than we actually think about, because these dips that we see or these declines that we see that get attention in the press are often bookended by really good years and then a really significant down year. And then you'll see that here in this plot. This plot is the stone crab commercial landings. This is data from Florida Fish and Wildlife going back to 1999 all the way up to 2019. And these are the total claws in millions of pounds. And if you look, you can see that you get a couple good years, but then on either end, those next harvestable years is lower. And that has become kind of the new norm. So you have another peak over here, but the prior years and the post years are even lower. And so we used to get 3 million to 3.5 million pounds of claws in the fishery, but now it's much lower. It's about a loss of over 1 million pounds in the fishery that we've seen in this time. It's about a 30% decrease in the overall harvest. So something's going on. So possible reasons for the declining in catch. So one could definitely be fishing pressure. We have 1.4 million traps in our waters during the season. In the off season, you can drive down A1A towards the Florida Keys, and you can see all the traps that these are lobster and stone crab traps, but you can see them all lining the median on the way down to the Florida Keys. But since the 60s, there's been a 100-fold increase in traps, and since then we've capped it off, and it's been stable. And there have been some fishermen that have gotten out of the business, but fishing pressure is definitely contributing or likely contributing to this. And this is from the last time this plot is from the last time that Florida Fish and Wildlife did a stone crab stock assessment. They're actually updating this right now. They're supposed to come out with the new one this year. But this was from 2011. And in 2011, they used these fishing mortality ratios and to calculate, doing some modeling to calculate a ratio, basically, to determine if the fishery is over harvested or not. And so the main thing I want you to focus on here is these last couple of years. So when this ratio, this fishing mortality rate to fishing mortality rate at the maximum sustainable yield is over 1, when that ratio gets over 1, that is when we consider a fishery overfished. And according to Fish and Wildlife's model, this has been an over-harvested fishery since the late 1990s. So another possible concern is changes in how we use the coastal habitat. These guys are a coastal species. So this is the range of stone crabs throughout their Atlantic stone crabs. We also have hybrids up in the Panhandle, and we have a sister species that is over in Louisiana's neck of the woods. But the Atlantic species is from the Carolinas all the way up into right around the Tampa Bay region, or maybe a little bit into the Panhandle. So this is the US Census Bureau's data of population increase in Florida. So you can see we got this exponential growth. Everybody likes to come to Florida. And if you look at where Floridians like to live, right, we don't really stay in the center of the state. We stay coastal, on the coastal areas. And so there have been some studies that have shown that increases in runoff and increases in organic nutrients can, in some of these coastal areas and on a seasonal scale, result in changes in pH that are 3 times faster than what we expect to happen for the end of the century. So that could be rapid changes in a few days or so after something like a rain event. So this work is from Toth et al. In 2018, and they report here that the impact that Florida Bay has on local habitats in the Keys. And so the Florida Keys is one of the main locations where we have the Florida stone crab fishery. About 90 to 95% of the fishery resides with what you see in this map. So along the bayside and somewhat along the bayside, and then up into the Everglade region or outskirts of the Everglades National Park over here is the predominant fishery. And what we see is that Florida Bay gets inundated with organics and nutrients. And so this plot is going to show you chlorophyll here in Florida Bay. So here you have red is high chlorophyll concentration and blue is the low chlorophyll concentration. And you can see that Florida Bay is getting these inimical waters that can be pumped or flow out through the Keys, especially in the Middle Keys, but also over towards they can even get out over into the western side of the bay where some of the fishery occurs. And those waters can also, at certain times of the year, be associated with rapid changes in pH. So most likely it's a combination of a lot of factors, but one of the things that for the fishery decline, but one of the things that we are not sure about here is no one has ever looked at what effects pH and elevated temperature have on their early life history. And so that's where my lab comes into play. The stone crab life history is similar to other brachyuran crabs. Excuse me. The males and females come together and mate. They mate during the winter. The females will store the sperm and fertilize the eggs when the temperatures start to warm above 27, 28 degrees. They then fertilize their eggs onto their abdomen, and a newly extruded egg mass will be this bright orange color that you see here on this female. They undergo a short migration. It's not very long. Most estimates think that they maybe migrate less than a kilometer, whereas something like a blue crab can migrate multiple kilometers to get offshore. And they release their larvae, and their larvae migrate or move offshore to complete development in offshore waters where they're away from predators, but also the conditions are more stable for their development. So when they develop their eggs, they eventually use up the yolk, and they get this kind of greenish brown hue to them. And the female, let's see if this works. The female will get up on her hind legs and flex her abdomen to help rupture the embryos. And what you see in that tank is all the newly hatched larvae, which if they survive, I'll play it one more time. The newly hatched larvae, if they survive, they'll be on your dinner plate in about 2 years from this point in time. So you can see the larvae with the naked eye. They're actually some of the larger crustacean, brachyuran crustacean larvae to work with, because they also only have 5 larval stages. Oops, didn't mean to do that. There you go. So as I mentioned, there's been some work done on ocean acidification or reduced pH on crustaceans. A lot of the work has been done up in Alaska and in the northern regions with the Tanner crab and the king crab. And those species show changes, increases in juvenile mortality. So we know that they're sensitive, the juveniles are sensitive. They also show decreases in their carapace size. So possibly some implications there for growth. We see intertidal species, which are often thought to be more tolerant to environmental extremes because of the environment they live in, also show sensitivity to changes in pH, where they, this porcelain crab shows decreases in survival by 30%, but also changes in their heart rate. So there's some physiological mechanisms at play there. Spider crabs can increase and delay their development time by over a month. And then even some commercial shrimp have shown there been significant decreases in body size. So the research questions that I'm going to focus on for this talk, there's 4 of them here that I'm going to go over today. So the first one is going to be talking to you guys about what the effects are of reduced pH on the stone crab's embryonic development, but also its hatching success. I'll then talk to you about another experiment that we did where we looked at simultaneous stressors, which were reduced pH, but we also added in elevated temperature. And we looked at their larval development and survival, so throughout their whole entire developmental sequence that they experience. Then we did a couple of behavioral assays to look to see if their behavior changes under these conditions as well, under elevated temperature and reduced pH. So I'll talk to you about some of that work that we did. And then at the end, I'll touch on some of the spiny lobster work that we did, where we looked at reduced pH impacting the spiny lobster's ability to discern settlement cues. Okay, so the first question that we are going to talk about is determining what the effects are on the stone crab's embryonic development. And so this was done a few years ago, and we had our ocean acidification system here, and we had basically 2 treatments. We had a control, which was close to ambient, 8.0, and we had a low pH treatment, which was 7.5. And so we acidified the water through carbon dioxide, adding in carbon dioxide mixture with atmospheric gas, and then monitored those tanks over time to make sure they were keeping with the experimental set points. Embryonic development in stone crabs has 7 embryo stages. The first embryo stage is 100% yolk developed inside the embryo. And over time, as they develop, the embryo changes color, but the embryo itself, you can see changes in morphology that happen. So one of the main benchmarks that we use to identify the larval or the embryo stage is the presence of the eye spot and also percentage of the yolk that gets consumed over time. And so as they get closer to hatching, their embryos get this dark green or brown color. They go from orange to rusty to this dark green brown color. The embryos, the yolk gets 100% used up. The eye spot becomes very prominent, and you can even see the heart beating, and you can see the abdominal tail segments as well. So this is their normal sequence of developmental stages. And what we did during this experiment was when we had a female that was early stage egg mass, we removed embryos daily, looked at them under the microscope, took digital images, and then we were able to measure embryo volume and development time. And we based it off of the 7-stage classification scheme that's been published. So this is what we found. So this is the time in embryo development. So going from the start of the experiment to the end of the experiment, and these are the embryo stages. And so here's the control. So this is the pH of 8, the ambient temperature. And so they took about 10 days to complete their embryonic development. And then when we compare that to the reduced pH treatment, we see that they were about 2 days slower to complete their embryonic development. It was about 24% slower development in the low pH treatment. And we compare this with a general linear model. And these results and the next couple of results we published in GEMBI. Because we got females from Fish and Wildlife that had early stage embryos, we didn't know how long they had those embryos. And so one of the things we did was we standardized the embryos by using females that were only in stage 2 of their embryo development. So that way we eliminated that prior duration or temporal scale that we don't have an ability to quantify from getting crabs from the field. So the next question is, does this delay in development or does the pH affect their hatching success? And so one of the methodologies that has been in the literature for some time now on looking at hatching success is to actually remove a pleopod with embryos and immerse them in the same conditions and put them on an orbital shaker to simulate or some type of motion device to allow them to simulate similar activity that the female would have from her aerating and pumping her abdomen. And then we wait until the female hatches. And once that female hatches, we wait until she's done hatching. And then we go back to the embryos and we look at the embryos under the microscope and we count the number of hatched to the number of embryos that didn't hatch to get an assessment of the hatching success. We were fairly confident when the females would hatch because we were looking at the embryos daily. So we knew when they were 24 to 48 hours, so we could prepare this experiment. And even though we were taking embryos daily, stone crabs hold about 300 to 500,000 embryos on their abdomen. So we would randomize where we took the embryos from, and we're only taking about 100 to 200 embryos max with each clump of embryos that we removed. And so over the course of the whole experiment, there were maybe, it was less than 1% of the entire egg mass that was actually being removed. So here is the hatching success. So this is the control on the left and the reduced pH treatment on the right here. And this is the median percent of hatching. So in our control treatment, we had really good success. Close to 80% of the embryos off the crab hatched. And when we compare that to the control though, we see this drastic reduction, about a 28% reduction in hatching success in low pH conditions. Again, it was a significant difference. We analyzed it with a general linear model. And so that suggests that there may be a potential bottleneck there for the population if they aren't able to acclimate or adapt to these reductions in pH. Okay, so with the results for the effects on their development and their hatching success. So we found that there is some effects and the hatching success is reduced. The development time is also slower in the reduced pH treatment. And I'll come back to talking about what the implications are for all these as we get done this whole section with the stone crab. So the next research question that we focused on was looking to see how reduced pH, so after they hatch, how does reduced pH and elevated temperature as simultaneous stressors impact their larval development and survival? So larval or stone crabs have 5 larval stages. So it's a nice crustacean to work with relative to something like the blue crab that has 7 larval stages. You don't have to, it's a little less, a couple less of the larval stages there to work with, which is nice. And they're actually larger than the blue crab and other crabs like the fiddler crab larvae. So these are their larval stages. You can identify them through the addition of different abdominal segments, for instance, like in this 3rd larval stage, they get an extra segment. And then by the 5th larval stage, they have these extensions on their pleopods or on their tail segments. So they're fairly easy to identify. And our experimental design, we used a fully crossed design here in this experiment. So we had in a water bath system that was digitally controlled, we had an ambient pH treatment at the summertime temperature, which was 30 degrees. We had a reduced pH treatment at that same temperature, 30 degrees. We then had an ambient pH treatment, but we increased the temperature by 2 degrees to mimic projections for end of century of what we anticipate is going to happen in coastal or tropical waters. And then we had a combined treatment with the reduced pH and also the elevated temperature. And so this system that we used was in collaboration with NOAA in the University of Miami. One of the nice components of this system was that this system was completely digitized. So we were actually able to gradually increase the temperature by a small fraction each day. And we were also able to increase the PCO2 over a small fraction each day. So that way we weren't just throwing these animals into end of century conditions. We were actually doing the best we could to minimize or gradually increase that treatment or to the treatment set points. So here are the days of development. It takes about 28 to 30 days for stone crabs to complete their 5 larval developmental stages. And these are the experimental treatments that we had with the mean PCO2 within each treatment. The blue represents the lower temperature and the red represents the higher temperature treatments. The triangles represent when we added or increased or treatments that had increased CO2. So the acidified treatments. And then the green symbol is going to be representative of the collection site. So we partnered again with Fish and Wildlife on this to collect crabs from the local habitat, and we were able to get some field samples of the site that we were working with. So here are the ambient conditions, the ambient pH conditions at both elevated and lower temperatures. And so they matched or within the range of the collection site. And as I mentioned, the nice thing about the system that NOAA had was that we could, over the first 5, 6, maybe 7 days of the experiment, gradually increase those PCO2 conditions and the temperature conditions so that there was no shock to the larvae, or at least minimizing the shock to the larvae. And then it stayed fairly stable throughout the rest of the treatment or rest of the experiment. So for some of these metrics, the first larval stages, we didn't have true treatment conditions all the way because we were slowly raising the temperature and slowly raising the carbon dioxide level. So one of the factors that we looked at during their embryonic development is their molt stage duration. And the molt stage duration is basically how long they stay within each larval stage. So here's their larval stage, the Z1, zoea 1, to all the way to the 5th zoeal stage, and their molt stage duration in days. And so the blue, again, is low temperature, red is high temperature, and you might be able to see it on your computer screen. The polka dots represent the CO2 or treatments or the low pH treatments. So even though we didn't have full experimental conditions in that first larval stage, we still see a temperature effect where it's accelerating their development. That was significantly different from the lower temperature treatment. That trend continued throughout their development. So stage 2, 3, and 4 larvae all had a similar trend. And actually the trend increased a little bit. They were about 1.5 days, molting 1.5, sometimes 2 days faster than the lower temperatures. So that was expected. We expected that the higher temperatures would be accelerate their metamorphosis and development. And then in the 5th larval stage, we still had the temperature effect on their accelerated development, but then we saw this delay in the 5th larval stage where they actually slowed their development by a day and a half maybe. And that was significantly different from the control. And so that has some interesting implications for survival that we'll talk about in a couple of slides. And so we did repeated measures ANOVA on this data set to look for those specific differences. We also were tracking a separate group of larvae. So we had larvae that were tracking for the molt stage duration, but then we had a separate group of larvae that we were tracking. And so same color scheme here, except black is the control. The reds are the high temperatures and the blue is the low temperature. And this is the survival probability. So we're looking at survival over their 30-day, 28 to 30 day development time. And the shaded region here represents the 95% confidence interval. So we had about 55% survive in the control treatment, which is pretty good for brachyuran crustacean. What we saw was the pH treatment had a different effect. We saw an increase in mortality under reduced pH. And then when we added in the temperature, and I plot these two together, we saw that the temperature treatments were not different from each other. Temperature was driving this decrease here. They were really getting knocked back and experiencing significant mortality with that elevated temperature. We analyzed this using a Cox proportional hazard model, and we found that there are significant differences. The low pH was different from the control, and then the temperature treatments, regardless of pH, were different from the control as well, but also different from the low pH treatment. And one of the nice things about the Cox proportional hazard model is that it gives you a likelihood that the animal would survive or die in that particular scenario. So using the hazard ratios, we were able to determine that crabs that were exposed to that reduced pH treatment were 1.5 times more likely to die than crabs in the control. But the big effect here was temperature. And so when crabs were exposed to that elevated temperature, they were 3 times more likely to die than the other treatments, which is pretty astonishing. So that is problematic for the fishery in the future without any adaptation, because these guys are already living close to their thermal range in the tropics. So the effect of reduced pH and elevated temperature impacting larval development and survival. So we did see effects, right? So we saw the effects on their development, and we also saw the effects on their survival. So now I want to jump into how do these changes translate to, or how do these environmental changes translate to, if any, behavioral changes? And so I want to define first a couple of responses that we look for when we're looking at larval behavior. So larvae can have a variety of responses behaviorally to environmental cues, but we kind of group those behavioral responses into 2 groups. One is a tactic behavior, and that's when the animal orients towards or away from a stimulus. So in this example, I'm using light. If the animal moves towards light, right, we consider that a positive phototaxis. And if it moves away from light, it's a negative phototaxis. You can also have changes in kinetic behavior associated with a response to an environmental cue. And changes in kinetic behavior are just a change in the activity level of the animal. So they might speed up movement, for instance, like you see in this cartoon, or slow down movement. And so we kind of develop experiments to discern what the differences are between tactic and kinetic behaviors to try to understand how these environmental cues might actually impact their swimming, because that's going to be important for the future of the population. So Sulkin proposed, he was one of the first to propose a negative feedback system for larval crustaceans. And so this negative feedback system is kind of a way that the crustaceans can regulate their depth. And so if you're a larval crustacean and you're moving towards the surface, you're going to experience environmental changes associated with decreases in salinity, decreases in pressure, and increases in light. Those environmental changes might stimulate corrective behavioral responses like a decrease in locomotor activity, a positive geotaxis or orientation towards the center of Earth, or a negative phototactic response, which then results in a response that's going to move the animal back down to a certain depth. As that animal moves down, they're going to experience the opposite suite of behaviors that you would see from when you moved up in the water column. So you get increases in pressure and increases in salinity and decreases in light. And as a result, they don't want to drift too far down, right? So they have these corrective responses, like an increase in locomotor activity or kinesis, a negative geotaxis to move back upwards, and a positive phototaxis to go towards the light, which would then result in the response of the animal going up. So these cues, sensitivity to these cues can change throughout development. And these obviously are not the only cues they can respond to. These are just a suite of them that are common. So I had actually done some of this work, some of this negative feedback model work when I was a master's student, and was able to characterize the larval transport or come up with a theoretical model for larval transport in stone crabs. And so stone crabs, when they release their larvae, right, we see the larvae go to the surface, and they respond through a suite of behaviors associated with responses to gravity, responses to pressure, and responses to light. So they have a negative geotaxis, which means that they're swimming up. They have a high barokinetic response, which means that as they experience increases in pressure, they swim up. And then they have a positive phototaxis, which means they also swim up. And they do not have an endogenous rhythm. So we know that the larvae, the first and second larval stages, are most likely up and near the surface of the water column. As they develop, and as they are at the water column or up in the surface, the ocean currents are favorable where they're going to help navigate them offshore to complete their larval development. As they develop into older larvae, they're going to get heavier, which is going to result in having a faster sinking rate. They reverse their response to geotaxis. They actually respond by swimming down, preferably, and not swimming up. They have a barokinetic response that when they experience a pressure decrease, which would be associated with going up to the surface, they actually respond by swimming down. And then responses to light in the later larval stages, they are negatively phototactic. So they change their sensitivity to these cues as they develop and get older. That positions these larger, heavier larvae deeper in the water column, where bottom currents can navigate them and move them back in towards coastal habitats where they will eventually settle into the juveniles. So one of the mechanisms that we wanted to look at was we wanted to do a quick behavioral assay to look at how this reduction in pH might impact their swimming behavior. And so to do that, we selected the GeoTaxis suite of experiments. And so during a GeoTaxis behavior experiment, we put the larvae in a dark room, we put them in a lucite chamber, we add in the experimental conditions, we time-lapse video record it, and then we have the animal in the chamber. We give them a period of acclimation in the chamber, and then we slowly and carefully turn the chamber and watch what the animal does. It's illuminated with far red or infrared light from behind. Then that allows the camera to see what the animal's doing, and then we can use the video to calculate their or identify their directional response, but also calculate their swimming speed. And so we did this during those ocean acidification and elevated temperature experiments. So on the x-axis here, we have the treatments, so the control, the elevated temperature treatment, the reduced pH treatment, and the combined treatment. And here I'm only showing this third larval stage. We didn't do the first larval stage because they weren't exposed to those environmental conditions yet, the treatment levels, because we ramped up the experimental scenarios. And these are negative numbers because these are going to be representative of larvae that were swimming downwards. And so in the control and elevated temperature treatment, we had about 30 or 40% of the animals swam down. So the majority of those individuals were swimming up in these GeoTaxis trials, which is similar to the prior results that I found during my master's. But when we exposed them to these low pH treatments, we saw a change in this behavior. They did not swim up. The vast majority of them swam down at around the 80% level. And so this suggests that they might be getting disoriented or just having difficulty or trying to get away from those environmental conditions or having difficulty sensing their environment. We also were able to use the video to analyze swimming speeds. And so we still have the same treatment levels on the x-axis, and the vertical axis now is centimeters per second, the larval swimming speed. And so in the control and elevated temperature, right, we see the vast majority of the animals swimming upwards or having a greater than 0.25 centimeters per second swimming speed. And then when we compare the swimming speeds to the low pH individuals, we see that the low pH individuals were swimming down faster and they were predominantly moving at a quicker clip to move out of those potentially corrosive waters. And if there was no corrective change in their behavior, if we just assume that they were going to continue to sink at this rate, this would be about 15 meters per hour that they would sink. So if we add this data into our conceptual model here of where the stone crab transport is and where it might be taking them, as you recall from that previous slide I had up there, the early larval stages like to be near the surface or likely near the surface where they're advecting offshore. The later larval stages are heavier and reverse their sign to those stimuli, those exogenous stimuli, and that results in them being pushed back towards nearshore habitats. But under this OA scenario, it's very possible that some of these earlier larval stages that we tested might be trapped or retained near the natal site due to the fact that they are reversing that behavioral response. They're swimming down at a faster rate, and so that might most likely will put them in more in contact with bottom currents than they would experience at the surface, which would keep them closer to where they were released. So we didn't see an effective temperature, but we did see an effective pH here. And so their reduction in pH disoriented them and resulted in an opposite response than what we saw in control conditions. Okay, so that is my story for the stone crabs. And so I want to talk to you real quick before I jump into the lobster story. I want to talk to you about the broader implications of the stone crab work because I didn't touch on this yet. So when we look at the embryos and the effects of these experiments on the embryos, it's potential here that there could be a bottleneck event with the reduced larval supply. So we're seeing about a 30% reduction in stone crab hatching success during exposure to low pH conditions. So if there's 30% less individuals, and we know that their mortality rate is very high at this stage, it's 0.00001% of them survive naturally. So this has potential to reduce larval supply and potential new recruits into the fishery. During the molt stage duration, under low pH, that fifth larval stage had a delay in molting. It slowed its development down by a day and a half, maybe 2 days. So a day and a half might not be biologically relevant, even though we saw a statistical response. However, whenever you keep yourself in the plankton as an zooplankton individual, you're exposing yourself to possibility for more predation. So it is possible that that delay might result as these heavier animals are moving back towards nearshore habitats. It's possible that they might experience additional predation by slowing that pH response or slowing the development to that pH. We saw lower survival, significantly lower survival under elevated temperature, but that's going to be an issue. It's going to be harder. We can't dump a bunch of ice into the ocean. It's going to be harder to mitigate that, but it's easier to mitigate changes in pH. So if we can reduce through local management, we can reduce some of the pH effects that we see in these coastal ecosystems. We saw drastically different sensitivity in mortality when the temperature was combined with pH than when it was just pH by itself. In fact, I would even argue that the 1.5 times more likely to die with low pH, they're most likely going to be able to tolerate that into the future over successful generations. But the temperature is going to be a limiting factor, I think. And then possibility of reduction in dispersal or transport into unfavorable habitats with those changes in behavior. So this could be problematic. We think that maybe the low pH might be impacting their statocyst because our other results show that there's no change in calcification. There's no difference in ash-free dry weight. There's no difference in dry weight. So they seem to be calcifying okay. However, the statocyst organ, which is associated with their antennae or behind their antennae and near their eyes, it's a calcium concretion that they use for navigation and orientation purposes. And so we propose that might be what's going on here, is that maybe that statocyst is becoming modified or degraded or impaired in some way. So if they can't migrate or navigate under normal conditions, it's possible that they could have difficulty expanding their range or moving away from these thermal extremes as they get warmer into the future. Which again would be a benefit for them, especially since they seem to be so sensitive to these higher temperature scenarios. All right, so my last section here that I want to talk to you guys about is this really exciting work that we did over the last 2 or 3 years, looking at the effects of reduced pH on spiny lobster post-larvae. So there's been some work from University of Florida folks in the Behringer lab that show that juvenile spiny lobsters are sensitive to reductions in pH, but no one had, until this work, had looked at the effects on their post-larvae. And so the spiny lobster fishery in Florida is another very economically important fishery. It's even more valuable than the stone crab fishery, brings in about $500 million a year. And they're harvested for their tail, and that's what ends up on the dinner plate. So here's an adult lobster, and the females will put the embryos onto their tail and brood them. After they release their larvae, they spend 9 months in the plankton, floating around the ocean. Eventually, they molt into the post-larval stage, which is what you see here. These clear, shrimp-looking organisms with these very large antennae. The antennae are almost a 1-to-1 ratio with the body size of the animal. And these guys are the ones that recruit back into nearshore habitats. They're incredibly sensitive to chemical cues. They basically ride the Gulf Stream until they get to the Middle Keys, and then they turn left and come into Florida Bay and settle. And that's the stage that we focused on, is this stage, where we collected them with plankton nets during nocturnal incoming tides, and then transport them back to Mote Marine Laboratory in the Keys, and put them into our experimental ocean acidification system. And we had half in a control and half in a reduced pH system. One of the cues that has been well documented in the literature that the post-larval spiny lobsters are positively attracted to is Laurencia. Laurencia algae is it's an algae that gives off these chemical cues that the post-larval lobsters love, and they migrate towards it, and they seek it out when they come back into Florida Bay. And so one of the research questions we want to focus on was, does the reduced pH affect their ability to orient to Laurencia cues? There's been a suite of studies that have shown different larval forms, from fish to some other crustaceans, that have some difficulty identifying positive settlement cues under reductions in pH. So to do this, as I said, we collected our larvae through plankton tows over a couple-year period. We added the larvae into our treatments, the ambient pH treatment or control, and the reduced pH treatment. So we only had 2 treatments here. The larvae are only in this post-larval stage for about 3 to 4 days. And so there wasn't a lot of time here to ramp them up. We did gradually introduce them to the treatments, but we couldn't do a similar ramp up like we did before with the other experiment. And so we had a seawater container that had no Q present, and then we incubated Laurencia in seawater for 24 hours. That was attached to a tube system that had peristaltic pumps that we could control, so we had the same flow. And then we exposed them to a Y maze, and basically we randomly selected a post larva, gave them an acclimation period within our chamber. This was all done in a dark room as well, and at night when they are supposed to be most active. Turned the pumps on and let the Q water do its thing, and let the control with no Q do its thing, and then monitor and watch see where the larvae went. And so what we did was we monitored attraction, and we monitored the time that the individuals spent as inactive. And so here's our chamber, and so we subdivided our chamber into sections. So if the animal crossed the Y threshold, the experiment was over. It was a success. They ended up in one side or the other. They made a choice. And then number 1 was where they were actually introduced and had the acclimation period. So we were able to quantify basically animals that made a choice, and also how long it took for them to basically get their engine going or get out of that section 1. So here is the proportion of larvae, and this is the ambient is in white, and the reduced pH is in black. And so we compared whether or not they were attracted to the Q. And so we did a binomial test here. We had 16 individuals that were tested in each treatment. And we saw significant attraction to the Laurencia in the ambient larvae. We had, of those 16, 14 individuals actually went to the Laurencia, and 2 of them ended up just wandering the chamber, or moving forward, but not necessarily going into the Laurencia chamber. We only had 2 animals in the reduced pH treatment that moved towards the Laurencia side of the chamber. We also monitored those that avoided the Q or didn't show any affinity towards it. And so we had 2 individuals that did this in the reduced pH treatment, and none of them avoided it in the ambient pH. The 2 that didn't make the choice were still on the Laurencia side of the chamber. That's how we defined that. And then no attraction or avoidance. We saw the opposite effect in the reduced pH treatment, where the vast majority of those individuals in the reduced pH treatment were not attracted to this to the Q or they avoided it. And we had this, we published this last year in Scientific Reports. The activity within the chamber. So again, we have the ambient larvae and the reduced pH larvae. And this is the time they spent before they moved out of the first section. And so we see the ambient larvae, it was about 100 seconds before they actually started moving. But we saw a significant increase here of the time it took for the reduced pH larvae to actually get out and past that first section. So the broader implications for this work, what we think might be going on here is that there is possibly damage to their receptors on their and their sensory abilities to sense that Lawrencia. And that could potentially result in inactivity due to the reduction in pH. And if that happens, they could also be susceptible to increases in predation, which could also lead to less recruitment coming in for the spiny lobster fishery. I want to quickly acknowledge some of the funding that we had for this. So my postdoc at Mote Marine Lab was funded by the Steinwachs Family Foundation. And some of this work, the stone crab work, was funded from a Mote Marine Lab Protect Our Reef grant that funded some of the larval development and embryonic development research. Florida Fish and Wildlife, the stone crab monitoring program, they helped collect animals and collect field samples. The spiny lobster work we did with Florida Fish and Wildlife as well, also colleagues at College of the Florida Keys, and then my collaborators at NOAA at NOAA's Ocean Acidification Program, and then my collaborators at Mote. And with that, I think I have a few minutes left to take a couple of questions.
Great, thank you. It's a
Yeah,
fantastic talk. So
yeah,
I haven't received any questions yet, but you guys can send them to me right now, and I'll facilitate the question asking.
Sure.
If you guys like. I have one question while we wait for people to write.
Okay. Yeah.
So there was one. So this is not really my area, so I don't even know how to verbalize it completely. But there was one experimental setup where you have a tank with a camera, and initially it's horizontal, and then it's flipped in the vertical direction. How does that work?
Yeah. So that system, that's the Geotaxis experiments, and so that is done. It's manual. We do it manually, and we do it. We drop the floor basically very, very slowly. It's a gradual change. We don't just flip it real quick because that would basically overturn the whole system. And we wait. They're very laborious experiments because we wait for the larvae to be central and not resting on the bottom. So that way we know that they're in the water column. And we also want to minimize the edge effect as well. So we don't use, if we do the experiment and the larvae is gliding along the side, we end up not using that because we don't know if the larvae is responding to the chamber itself or if it's responding to the cube. So we try to minimize that the best we can by taking our time doing it and doing it very slowly. But we just haven't had the funding yet to do that digitally, set up a little gear system to do it. Yeah, good question. That's traditionally how they've been done. These negative feedback model experiments for Geotaxis, they've been done that way for the last several years, or even beyond that, actually, before I even started.
You also mentioned using an infrared light to get the video of the larval movement in the chamber, right? Mm-hmm. So is that because if you use like visible spectrum light, it'll impact their behavior?
Right. Yeah. So they do have a phototactic response, and so if you use regular, visible spectrum light, they see that different wavelengths. But the crustaceans are they're not sensitive to the reds. And so you can use the reds to illuminate your darkroom without any worry about the behavioral impact of that. Forward did this work at Duke University. He kind of described those behavioral responses relative to light. He was one of the first ones to do that, and his work consistently showed that the red spectrum is they don't have any impact or sensitivity to. Yeah, good question.
So we have another question from Fabio Del Piero, who's asking, do you think this Mediterranean species may suffer the same issues? And the species that they're sharing is Cancer pagurus, and they're asking, like, maybe it could be more resilient, less resilient. What do you think?
Yeah, let me click the link.
Oh, you probably got it too, right?
Yeah, I just saw it. Yeah, good question. I don't know, possibly. So the crustaceans are a weird group when it comes to acidification, and it's because we see different sensitivities for different species. So they're more species-specific responses. And you know, when some of the OA studies we've done with the adults, we don't see much of an effect. Juveniles, we've seen variable effects. And I think it depends on the stressor that they might be coupled with. But even the larvae seem to be resilient to the OA in the sense that, you know, they might have more of a mortality effect and they might have a little bit slower development, but they're still calcifying and they're still surviving. I think temperature is the main factor that's going to dictate or drive the response here. But yeah, with the cancer crab, Cancer pagurus, yeah, I'm not 100% sure if there's been any additional studies looking at them and what their effect might be.
I've got a quick question.
Yes.
It's kind of a loaded question, which is sort of based on some potential collaborations that Phil and I are hoping to do.
Yeah.
You know, a lot of these experimental treatments are sort of mean conditions, right? What do you think, you know, these coastal environments are heterogeneous in space and time. And so what do you think about what would happen to these animals if they're moving in and out of sort of deleterious conditions? Because you mentioned that it might be the degradation of some sort of you know, structure in their bodies. So I'm just interested in how heterogeneity might play a role. Yeah,
so that's a great question, because we do actually see variability in the pH in their habitat, and so we're getting better at it, but it's a little bit difficult during these OA experiments to fluctuate that. But there's been technology that's been developing that we're moving towards that can actually shift the pH on like a diurnal cycle, for instance. And that's where we want to go is to look at that effect and identify if that variability exposure is playing a role. And so so this is a really good question, and I want to go to this plot because we're actually starting to look at that right now, and hopefully we'll have some data after this summer. So we see a reduction under those mean conditions, but what we also saw was that there were some individuals here that had 80% hatching success, which was very similar to the control, and then we had some that were just not good at all at releasing the larvae under low pH. And so what we're actually testing this summer is this exact question, is looking at crabs that are being conditioned in habitats within Tampa Bay that have variable pH and that are more stable in pH. So like a mud sand habitat versus a seagrass habitat. And we're conditioning them in the water we're putting them in the water, we're letting them release their eggs, we're feeding them, every day we're waiting for them to put out a new egg mass, so that way they're undergoing embryonic development in those conditions, and then we're bringing them back into the lab and we're looking at how that might impact their success. And so my guess is that the crabs that have that variable experience, to kind of get to Dan's question, is they're going to be more successful, is what I think is going to happen, and they'll have a better time hatching, they'll have a better survivorship, and I think that we won't see the effect that we see from this experiment during the low pH conditions. So I think that prior exposure is going to be really important. Yeah, it's a good question.
We have two more questions here. One's from Chris Madden. They're asking, might cycles of drought or a high freshwater flow affect recruitment success? Your early graph might have shown good success in the saltier years.
Oh, I'm sorry, what was the last part you said?
So in one of your early graphs, you mentioned that you'll have like a few years where there's a good harvest, and then there'll be a year that's really low. I think that Chris Madden is saying that it could be attributable to cycles of drought or high freshwater flow.
Yeah, there's no correlation with those that we know of. We looked at that. We looked at red tide as well, and seeing if some of those years correspond to red tide and if that results in the harvest. And the only spot in Florida where we see a correlation with red tide is in the southwest corner, the Naples region. We see a 60% decrease in harvest in that region. But those other locations, we don't see any of the harvest attributed to changes in salinity or drought.
Ben Farmer has a question about crustaceans and disease. They're asking how that might interact with the pH and temperature stressors to inhibit larvae.
Okay, so are you talking about the Panulus argus virus? Is that what they're referencing?
Ben, if you'd like to speak up, go ahead.
Yeah, I just know there's a lot of diseases, especially in lobsters, that they're struggling with.
Yeah, that's a great question. So those are the next stages. We want to basically expose them to disease as well, add another treatment in there, and look to see what the susceptibility to that disease is under these conditions, and if they can still sense it. So the juveniles are excellent, and some of the adults are excellent at sensing the disease, and they actually the spiny lobsters aggregate. And they will actually shun individuals or eject individuals from the population or from that aggregation that are diseased. They can sense it through their urine, hormones in their urine. There's been some work that's come out that has shown that there might be some impairment from the Behringer lab at University of Florida. They've done some of the disease work to look to see what the effects are. And I think they're still doing additional studies too, but we don't have a firm answer yet on how pH might interact with the disease prevalence in the Florida Keys. Yeah.
Cool. Thank you.
Yeah.
Okay. Any last second questions? Hit enter. And otherwise, I think we'll just have to thank Dr. Gravenese for coming. Virtually, of course.
Yeah, it would have been cool to be there in person. But
yeah, maybe in fall if you have some more things to show.
Yeah. Do all the red tide and hypoxia stuff that I didn't talk about.
Yeah, that'd be great. Okay, so I don't think we have any more questions. So thank you.
Cool.
I'll end the meeting now.
All right. Well, thank you guys. I appreciate it, and it was fun. So thanks a lot
Take care.
for the invite.